So first, thank you for joining us this afternoon. The goal for this presentation is to give everybody a better understanding of cell counting. I know for myself, in particular, maybe, that working in the lab and doing routine cell counts, it was kind of a part of the process that I would sort of skip over, try to rush through it, get it done quickly. Um, But when you think about it, when you're doing a cell-based workflow, you really have two inputs into that workflow. You have your treatment, whatever it is, a drug or a virus or something like that. And then, of course, you have your cells. So the cells make up half of the inputs into your workflow. And if what you're putting into the workflow, if the count cells per mil isn't accurate or at the very least consistent, then all of your downstream experiments are going to suffer from that. So, kind of the goal today here is to give everybody an overview of cell counting and then just a few things to keep in mind while you're doing cell counts, so that the process will still be quick, but hopefully it will be more accurate as well. So here's the overview of the presentation today. I'll background on how cell counting works. I assume that everybody kind of already has a good idea of this, so that'll be brief. And then we'll get into the 7 tips for improved cell count accuracy. And then at the end, we'll have a Q&A session. And I think Rowena mentioned this, but since we have a fairly small group here, if as I'm going through a slide, if there's a question, feel free to just maybe jump in or throw something in on the chat at the end. And I think it would be easier to address it right there and not have to go back later and readdress things. And since we have a small group, I hope it won't interrupt the flow too much. So we'll see. So you have 2 basic methods for cell counting. Your first is flow-based, the second is image-based. Flow-based, we're talking about flow cytometers, culture counters, something where you're running cells through a microfluidic and it's being counted either by fluorescence or by impedance or something like that. These methods are great for fast counts of many cells and then cells within a population. So, looking for different types of cells within that population of 1 million cells based off something like size or fluorescence. They are, of course, expensive and laborious to run. And if you want to get quantification from them, you either need to make sure that you calibrate your pump regularly, your fluidic pump, so that you know exactly how many mils are passing through per second, or you need to run a standard with your cells. What we're going to be focusing on today is image-based cell counting. And that comes in 2 flavors, manual hemocytometer, which is what everybody knows, the grids that you look at under the microscope and you sit there and click away on your counter, counting cells. And then automated image-based hemocytometers, sort of like the cell drop. Those would be lower throughput than if you're doing a flow-based system, but they will give you accurate and cells per mil viability, GFP transfection percents, that kind of stuff, very quickly and a lot more cheaply than using a flow-based method. So here's the old school manual hemocytometer. This is probably most of the people here. Certainly gives flashbacks to me. But I mean, basically the concept is that you have a fixed area and then you count the number in that area. And most people do something that's 1 millimeter by 1 millimeter with a cover slip that holds you to exactly 100 microns. So you have, I believe it's exactly 10 microliters under that area. When we're talking about automated image-based hemocytometers, it's exactly the same principle. There's no additional magic going on, no extra sensors or anything like that. You're just looking at a number of particles under a field of view and counting those particles in a fixed volume. Three key differences, though, from the old school manual hemocytometer. One is you have a much larger field of view. In the cell drop, for example, it's about 3 times bigger than what you would count in a manual hemocytometer. So right away, you get a more representative look at your sample. Um, Also, you can measure your samples much more quickly. So instead of taking a few minutes to click through 200 cells and count them, you can do that in a matter of seconds on an automated hemocytometer. So that opens you up to being able to run a few replicates of your sample, maybe get a more representative look at what you've got in the tube, again, giving you more accuracy. And third, you get consistency with an automated hemocytometer. So different people count things slightly differently. That's just the nature of something that's subjective, like cell counting. So what I might call a live cell, you might think is a piece of debris. On Friday afternoon, when you're rushing to finish up, you may speed through the count, maybe not get the most accurate count. When you have an automated hemocytometer with fixed protocols, you get consistency every single time. And we've done a few studies on this. Other people have done studies on this. There's some literature available out there that just shows you have much better consistency, much lower standard deviations when you're looking at an automated hemocytometer compared to manual counting, especially when you're looking at it person to person. So our 7 tips for accurate cell counts: cleaning, mixing, making sure that you minimize cell clumping, choosing the right Brightfield or fluorescence in this case, optimizing your instrument settings, using an appropriate chamber height or an appropriate dilution, and adjusting your count parameters. That one we'll go into. That's just making sure that you're checking the counts on an automated hemocytometer and dialing things in properly. So tip number 1, cleaning the sample surface. If you're talking about a manual hemocytometer, you just wipe away the sample with a Kim wipe, apply some ethanol to the surfaces, either on the Kim wipe or load it separately, wipe it again. And then when you're done, you put on a fresh cover slip, put it in the case, put it back on the shelf. Easy. With a disposable slide-based hemocytometer, you need to discard the slide when you're done. We've heard and had people ask this question of what's the best way to pull the slide apart and then clean it and be able to reuse the plastic disposable slide. There's no good way to do that. Do not do it. It doesn't work. You won't have accurate data if you do that. If you're talking about the cell drop, so an automated hemocytometer with reusable surfaces, you just wipe the surfaces clean, and then after the final sample, you want to load 10 microliters of ethanol, just to make sure that for the next person who goes up there, it's clean, and then you're done. So something that there seems to be a little bit of confusion about when I've been out in the field doing demos is how crucial is it when you're looking at the live preview before you've loaded cells that it's totally clean. For the most part, if you have a piece of dust on there, it'll just get flushed away. And I just have a quick example video here. You can see the particle in the lower left, the bit of dust, that as you load cells, just gets flushed off the surfaces. This is true in most cases, unless you're talking about something that has dried onto the sample surfaces. So if somebody left a previous sample on there, it got dried on, then you do need to do some additional cleaning. Tip number 2, make sure that you mix well before loading. So I think this one is kind of obvious. Everybody knows you need to mix your cells. But I think what's maybe not so obvious to people is how quickly the cells can form a gradient in the tube. So the recommended protocol is to either vortex it right away and then measure. A lot of people don't like to vortex their cells, which is understandable. So they like heading what's in the tube up and down a half dozen times before they load their sample. Um, so that's the recommended way to do it. If you wait, and you'd just be surprised how quickly the cells can settle to the bottom of the tube. So I just did a quick experiment, took some beads, a target concentration of 1.5 million beads per mil, mixed them well, loaded them onto the cell drop. And then I did the same thing, but then I waited about 15 seconds from after I mixed the beads to when I loaded them on the cell drop. Kind of thinking if you were to get distracted, if someone were to come into the lab and ask you a quick question, or were to look down on your phone and see a text message, this is going to distract you for about 15 seconds. And at least I previously would have thought that was fine to just go ahead and load my cells. But what I found was when you look at the well-mixed sample, you get about 1.5 million cells per mil, about at target concentration. When you have that same sample that's been mixed, and then you wait about 15 seconds, and then take something from near the bottom of the tube, the different people load from different areas of the tube, you go up to about 2.5 million cells per mil, just from 15-second wait. And then when you sample that same tube, mix it, sample from sort of close tube, you go down to about 600,000 cells per mil. So I was pretty surprised that you get a 5x difference between the bottom of the tube and the top of the tube pretty consistently after just waiting for 15 seconds. So, this is one of those things that might seem obvious to people, but I think that a lot of times it can have more of an effect than might be realized, to make sure that you're really mixing well and loading your cells onto the hemocytometer right away after the mix is done and not getting distracted. Okay, tip number 3, minimizing cell clumps. So to get an accurate cell count, you need a representative sample from what was in the tube, or really you're looking at what was originally in your flask or plate. Clumps mainly come from people not being patient enough when they're doing the trypsinization process. It could also happen if you have a culture that's really overgrown, so you've let it go a couple of days too long, and you just say, I'm going to go ahead and use this anyway. So when you get a clump, it misrepresents the sample, because if you have a big clump, it can completely bias the count to be very high, instead of giving you an accurate representation of the actual count. But they're also very difficult to count manually. So you end up guessing more. I mean, at least I know if I were to look at a poorly digested sample like this with a bunch of big clumps, I might start counting it and then say something like, okay, this one here in the bottom right, that's probably about 25. And that one's sort of in the lower middle, that one's probably about 20. And not really take account, just because it's difficult to keep track of where you are in that clump. Versus if you have a properly digested sample, like shown here, you still have a few clusters where you've got 2 or 4 or 3 cells mixed together in a cluster, but that's pretty easy to count, both manually and for an automated hemocytometer. Of course, the automated hemocytometers, like the cell drop, have clumping algorithms in them, or declustering algorithms, as we call it, that will do a pretty good job of getting you an accurate count of what's in that cluster. But the problem is that you're not getting a representative look in the field of view of what is in the tube if you have a couple big clumps of cells in there. So to kind of prove that point, I did a little experiment of clumping cells. I just took two flasks, seeded them exactly the same way, let them incubate and grow for the exact same duration. And then I poorly digested one flask with my trypsin. I just let it digest at room temperature, left it in the hood for about a minute, and then took the flask, smacked it with my hand to try and dislodge anything, and then I added my media. The other one I digested according to protocol. So I put it in the incubator at 37°C and let it sit for 3 minutes before I pulled it out. And I did 5 replicates of each one on a cell drop. What I found was that on average, I ended up with about the same number, which was encouraging. But for the poorly trypsinized flask, we're talking about the one that looked like this. I had a huge standard deviation compared to digested flask. So I think my standard deviation was 10 times higher for cells that were trypsinized incorrectly. So it kind of then becomes, did you get, most people are going to do one count from their flask or from their plate. They're not going to do 5 counts to get a good average. So did you get something from the top of the standard deviation, or did you get something from the bottom of the standard deviation? That's going to have a big effect on your input into your later experiments. Okay, tip number 4, making sure that you choose the proper staining technique, bright method versus fluorescence for your live dead cell counting. Depending on where your sample is coming from, doing a Brightfield, so trypan blue, versus a fluorescent stain is really, really critical to make sure that you get an accurate count. So primary cells are notoriously difficult to count with your trypan blue stains. That's because even if you do an isolation, you're still there's almost always some red blood cells contaminating the isolation. You have other debris from the isolation. You have plasmids that could be possible. Those are all going to show in the background. If you're manually counting it, it's almost impossible to distinguish unless you're really, really highly trained. Is that a red blood cell? Is that a dead cell? Is that a live cell? You just don't know. So if you're collecting primary samples, so from a ground tissue or from whole blood, that even if you've done an isolation, you really need to be using fluorescence for maximum accuracy. So when we're talking about fluorescence in live dead cell counting, we're talking about acridine orange and propidium iodide, or I'll be calling them AO and PI for the rest of this presentation. So you combine those two dyes together. They're commonly available. They're cheap. They're really not any more expensive than buying trypan blue. And AO passes through the membrane of all cells, and it stains the nucleic acids in that cell's green. They fluoresce green. PI can only pass through the membrane of a compromised cell, and that stains all the nucleic acids in that cell red. If you have a dead cell, both AO and PI are present, or a cell with a compromised membrane, both AO and PI are present in that cell, but PI quenches the green signal from the AO dye. So if you have a green fluorescing cell, that must be a live cell because PI was not there to quench the dye. And if you have a red cell, that must be a dead cell. So here's an example of a primary cell sample that we counted in our lab. I believe this is PBMCs, so peripheral blood mononuclear cells that were isolated from mouse whole blood. And you can see a bunch of green cells glowing very nicely, very clear that those are live cells. There's a few red cells as well, but then also behind that, you can see all kinds of other things. A bunch of dark spots, those are probably cells. A lot of other things back there, who knows what that is. But they're definitely not live or dead nucleated cells. If I count that exact same sample with trypan blue, now you see a very different result. The total count is much higher. You have more both live and dead cells present. So your viability is going to be off. You might think that your culture is not very good, or your primary cell sample has a lot of death when really it doesn't. And your total cell count, your live cell count, your basic inputs into your experiment are going to be off as well. So you're going to actually have much fewer cells than what you had thought you had. So we have done some studies on this. Other people have done studies on this. There's literature available that trypan blue overcounts in our hands by a factor of 2 when compared to AOPI when you're looking at a primary cell dilution. So lymph nodes, blood cells, you're overcounting by a factor of at least 2 with trypan blue. When you're looking at tissue culture, you see much less of a difference. You see good agreement between the two methods with tissue culture. And these examples here, the tissue culture are CHO, and the primary cells are PBMCs. And we just did dilutions, verified them on a culture counter, and it's really startling to see the difference with the primary cells of how different a count that you get between the two methods, with the AOPI being verified to be more accurate. Okay, so tip number 5, make sure that you optimize your instrument settings. So when we talk about instrument settings, we're talking about things like focus and exposure. Making sure those are correct are very important to get your accurate concentration and viability data, especially when you're talking about trypan blue. Trypan blue is very vulnerable to poor focus issues. Live cells may appear dead. Dead cells could appear live if the focus is not correct. Fluorescent techniques tend to be a little more forgiving, but good technique is still important. That's something that we hear a lot from customers. Like, oh, they'll send us images, they'll say our counts aren't right. And we'll look and their focus is out, and they think that because it's fluorescence that it shouldn't really matter that much. But as we'll see, look at some examples here, it definitely does matter, and it's fluorescence as well to make sure that you have good focus and exposure. So here's an example of a well-focused trypan sample. So you have sharp outlines around the cells, and you have bright white centers, and those you get a good focus. And you see with this well-focused sample, you have a viability of about 90% and live cells per mil of about 850,000. That exact same sample, just tweak the focus out a little bit, just 1 or 2 clicks the wrong direction. You still have some bright centers here in some of these cells over to the right there. So, it is possible that somebody might look at this and think that the focus is, but your viability now has gone down to about 60%. So you went from 90 to about 60%, and your live cells per mil went from around 900,000 to about 600,000. So you're going to have a very different calculation and a very different amount of cells that you're going to be adding into your experiment based on how you focus this. Exposure is also very important for trypan blue. Here you have a correct exposure, so you have a nice gray background, you have bright white cell centers, sharp dark outlines around the live cells. And here you got a viability of about 85% and live cells per mil of about 600,000. that same sample with the exposure set too high, your viability has now gone up to about 93%. But the reason that I chose this particular sample is you see sort of here and here where you previously had two things that obviously were bubbles and weren't being counted. Now those look an awful lot like dead cells. So what happens if your exposure to is too high is that tends to hide a lot of things, potential problems with your cell culture, like if you have bacteria contamination. Um, in this image here, you're going to very clearly see little bacteria swimming around. In this one, they're going to be totally washed out. You're going to think that your sample's good, and you're going to move on with your experiment, not knowing that you had bacteria contamination. So moving on from trypan blue now into proper focus for AOPI. Here is a properly focused Brightfield image. Here is the exact same image properly focused in the green channel. So you see some nice bright sharp cells fluorescing green there. And here's your count of that sample. You got about 92% viability and about 900,000 live cells per mil. So that same sample, now I just adjusted the focus a little bit. It's now fuzzy outlines. And like I said earlier, a lot of people think that for fluorescence, focus really isn't that important. I'm not sure why. That's just something that we've heard a lot from customers in the field. That just seems to be a myth that's going around. But what you see now, you have your poorly focused Brightfield cells. They're not terribly out of focus. But when you go to the green channel, you now see that those cells are very dim and very diffuse. So it's not a clear, sharp, bright fluorescing cell anymore. And when you take your count, you've now gone from about 93% viability down to about 77% viability. And your live cells per mil have gone down to 1 million live cells per mil. So just by changing the focus and the exposure a little bit, I was really able to change the count and therefore your input into your experiment. So very important to make sure that those settings are done correctly. Okay, tip number 6, using the appropriate dilution, or in the case of a cell drop where we have adjustable chamber heights, being able to adjust the chamber height properly. So Hemocytometer manufacturers recommend between 100 and 200 cells in a square, 1 by 1 millimeter square. I I didn't know that until I looked it up for this presentation. I don't think anybody follows that recommendation. But of course, it is important to make sure that you don't have 2 cells per square or 2,000 cells per square. Otherwise, you're not going to get an accurate count there. For the cell drop, we actually have 3 different chamber heights that you can use. So there's a 100 micron chamber height. That's the standard one. That's the same as the manual hemocytometer. That's what most disposable slides use. And then we have a cell density that we recommend in that chamber height. And then if you're too diffuse, if you don't have enough cells at the 100 micron chamber height, what you can do to get a more accurate count is go up to a 400 micron chamber height, load 40 microliters of the sample. Now you have 4 times the number of cells in the field of view, giving you a more accurate count of that sample. If you're way too dense, you can lower down to a 15 micron chamber height, load 5 microliters of your cells, and now you've essentially done a one-to-one dilution on that cell without having to actually go and physically do the dilution. So what you can see, if you have cells at 100 microns that are way too dense, like what I have here on the bottom left, or way too sparse, like I have here at the bottom right, like see like you only have about 5 or 6 cells there. You can just adjust those chamber heights. And now on the bottom left, essentially doing a 1-to-1 dilution on your cells. You now at least can see some background on those cells. It's still probably going to be pretty tough to count, but it is now possible, unlike with the previous image. And then with your sparse cells, you now have about 4 times as many cells available to count. And I think what I was trying to show here is in that previous image, you only had maybe 5, 6 cells in the field of view. Now at 400 microns, you can see there's a lot more cells, much more than 4 times more. And that's because you've gotten just a more representative sample. So you're going to get a more accurate count now from that sample without having to go and spin down your cells, redilute them in a smaller volume just to get an accurate count before you decide how to proceed with your later experiments with those cells. Okay, so the final tip here is making sure that you adjust count parameters. Unlike the other tips, this one really only applies to automated hemocytometers. So automated hemocytometers do have some advanced software. They're able to do image recognition on their own, but they do require the users to be the final experts on the cells, to look at the counts that are coming from default parameter sets that are loaded on the instruments when they're shipped, and tweak those default parameter sets and set what we call protocols for particular cell lines. So DeNovix cell drop has 3 main parameters that the user is able to adjust to fine tune their cell counts. Those are minimum, maximum diameter. So that's just setting a size gate for what is and what isn't a cell. What we call roundness. We'll get into a little bit more detail about exactly what that means, but that's kind of a score of how good of a cell an object is. And intensity threshold. So that's for live dead counts. How dark does something need to be stained to be counted as a trypan cell, or how brightly does it need to be stained to get counted as a fluorescence cell? So the min-max diameter is pretty straightforward. There's one thing that we have on the cell drop that I think is kind of nice is that you can do real-time size gating. So you can either look at a graph of your cell diameters, which you have here, and say you happen to know that these are yeast cells and they really shouldn't be over 8 microns in diameter. You can adjust it like that. You also can visually adjust things on the fly. So you can see cells coming in and out of the count in the image and in the data on the left. So that's just a nice feature that the cell drop has that most automated counters don't also have. That you can change that on the fly. You can visually look at it and dial in the count at the same time. And then once you've got it dialed in, you can save it to your protocol. So the next parameter I want to talk about is the thresholds. So how dark something needs to be counted as a cell. Our default protocol for trypan blue, the threshold is 35. So that's sort of on the lower side, 50% being right in the middle of the grayscale. So that's looking to say how dark do you need to be counted as a dead cell. And here in this first image, the default protocol found about half the cells were dead. And then you had about 250,000 cells per mil. Changing that protocol, so bringing the threshold way up to 80. So something has to be quite dark on the grayscale to get counted as a dead cell. Your viability percent now goes to almost 100. just by changing that one number, and your cells per mil gets cut in half. And then bringing that way down the other way, down to 10, your viability goes down to 37%, and your cells per mil goes way up. So a lot of things are getting counted as cells now. This might seem, really basic. Why not just leave it at the default? But different cells tend to stain differently. Some cells may stain very, very darkly, so you want to set that threshold to be kind of high, so that make sure that you're not counting any debris if you have debris in that sample. Some cells tend to stain very lightly, so you might need to set that threshold down around 10, if you want to be getting a good viability count on those cells. So it's really up to the user to understand that parameter and adjust it correspondingly. So the next parameter I want to talk about is what we call roundness. So I mentioned that's a score of how much something looks like a cell to our algorithms. And what it's looking for there is how basically circular is the object, how much of a circle is the object, and how complete is the outline around that object, to get counted as either a live or a dead cell. So again, the default parameters, your live roundness is 50, so kind of right in the middle, and your dead roundness is 25. We tend to send that one lower for the default because dead cells tend to be less regular. So with the default protocol, again, this is the same sample, viability 48%, cells per mil, about 250,000. When I bring that live roundness way up, I'm now biasing things away from being counted as live and biasing them towards being counted as dead. So my viability went way down to 8%, and then going the other live roundness down to 10, your viability goes up to about 100%. So, the users do have a lot of control with these roundness settings to say what gets counted as a cell or not. So just to summarize that, default parameter sets on automated cell counters are really meant to be a starting point, something that we want the user to take a few cell counts on different cell lines with, and then play with those settings. I will say if you're doing tissue culture cells, something like a CHO cell or a BHK cell, the default parameter sets are usually going to work pretty well. But for different cell types, fibroblasts, things that might be irregular, primary cells, you're certainly going to have to adjust those parameter sets. DeNovix has recommended parameter sets on our website for different cell types, and those are available to go get the parameter set, load them on. Say if you're using a MRC5 cell or whole blood or something like that, we have published recommended parameter sets. But still, those are meant for you to check us on, not just to be taken as truth. So the users really need to understand how the parameters work, what they do, how to fine tune them. Once parameters are set for a particular cell line, you don't need to change them regularly, but I do still recommend that you check occasionally, make sure that somebody hasn't gone and edited your parameters, and make sure that you're still getting accurate counts for that cell line. Okay, so that is the summary. Here's the summary. Proper technique and good understanding of parameters, things like exposure, and focus are key to getting accurate cell counts. Improper cell counting really does have downstream effects. You can end up having inconclusive and conflicting data. on your experiments. If you don't make sure that the input that you're putting into that experiment is good. So understanding these 7 key tips, they're simple things. Um, Really, once you know them, once you understand them, they kind of become second nature. But they can greatly affect the consistency of your research, leading to much better data